Hello there everyone, and welcome to this Biology Mind A-Level tutorial on DNA structure and the double helix. So, in our last lesson we learned about polynucleotides and their formation and breakdown. You now understand that multiple nucleotides bond with each other through condensation reactions to form a polynucleotide. And this involves the formation of a phosphodiester bond, and they can then be broken down via a hydrolysis reaction. So we have two key learning objectives in this tutorial. Number one, to talk about the structure of DNA, and number two, how we can purify DNA. So a single strand of DNA has two different ends, and due to the orientation of the deoxyribose sugar, there are two ends. So the starting point of a single DNA strand is known as the 5' end, which you can see here. And the end point of a single DNA strand is known as the 3' end. So that means that the DNA runs from the start at the 5' end to the end at the 3' end. So this would be 5 and this would be 3. And the other strand of DNA in the double helix runs in the opposite direction. So 5' is now down here, and the end of the strand is the 3' end, which is why we refer to them as antiparallel strands. So, as you know, DNA is composed of two complementary polynucleotide chains. And as I said, the chains run alongside each other and are antiparallel because they run in opposite directions. So one strand goes from 5' to the 3' end, and the opposite strand goes from the 3' to the 5' end. And hydrogen bonds hold the polynucleotide chains together, and they form between complementary base pairs on two nucleotides. So you can see the hydrogen bonds here form between complementary base pairs between the two strands of DNA. Now let's just remind ourselves of the complementary base pairs. So adenine forms a complementary base pair with thymine, and you can see them here. So this is adenine and this is thymine, and this is the complementary base pair. Don't worry, you don't need to memorize these structures. And then guanine forms a complementary base pair with cytosine, also through that complementary bonding. So it's those hydrogen bonds that are formed there. And in this complementary bonding of base pairs, a DNA double helix is formed. And the two antiparallel polynucleotides chains twist to form this helical shape. So you can see that this is one chain and this is the second chain, and they're running in opposite directions, remember, from the 5' to the 3' end. Now, the nucleic acids from the DNA at different biological samples can be purified through a reaction that we call a precipitation reaction. So this involves breaking down the cells in the sample and causing the DNA to precipitate so that it can be separated from the rest of the solution. So let's talk about how we can do that. So first of all, we add detergent and salt to the sample solution. So the detergent breaks down the lipid membranes in the sample, and the salt neutralizes the charge in the DNA's phosphate backbone. The solution is then heated to further break down the DNA, so the high heat denatures enzymes in the cells as well as breaking down the DNA further. Now, protease enzymes, which break down peptide bonds, are added to the solution so that any proteins bound to the DNA can be separated. And ethanol helps the DNA to precipitate because DNA is not soluble in alcohol, and the ethanol is an alcohol. Therefore, adding ethanol to the solution allows the DNA to precipitate on top of the solution and be separated from the rest of the cellular material. So that is our tutorial complete for today. Well done for following through, and I'll see you for the next one.